Hi everyone, welcome back to my channel. In today's lesson, we are going to look at factors that affect enzyme action. We are covering content for the AS Cambridge syllabus. Yes, code of biology is 9700. So let's dive right into the factors that affect enzyme action. We are going to look at 4 factors. Excuse me, we have temperature, pH, enzyme concentration, and substrate concentration. So let's start with the first factor, which is temperature. The effect of temperature on the rate of enzyme activity. And we are going to use this graph to describe how temperature affects the rate of reaction of an enzyme. Remember, when we are looking at rate of reaction, we are looking at how fast or slow the speed at which an enzyme is working. Sometimes the enzymes are working slowly, catalyzing the reaction slowly. Other times they are doing it very fast. And temperature is one of the factors that determines whether the enzyme is going to be fast or slow. So you could be told to describe such a graph. And whenever you're describing, you look at, are we having an increase? Are we having a decrease? Are we having a peak? Are we having a constant? When you're describing, you're just stating what you are observing on the graph. You do not need to explain. So when we are describing this graph, we can see that at low temperatures, like an example at 0 degrees, the rate of reaction is really low. Now we see as temperature increases, if you look at the temperature increasing from 0 to 10, here at 10, when temperature increases, we see the rate of reaction also increasing because it's higher. When the temperature increases to 20, we still see the rate of reaction increasing. And the rate of reaction increases with temperature up to when we have a peak obtained at 40 degrees Celsius. At the peak, that is where we have the highest rate of reaction, the maximum rate of reaction, the highest rate of reaction. And the moment the temperature increases beyond 40, we see the rate of reaction is decreasing. Now, this is an exam tip. Whenever you enter into an exam and you're told to describe, please provide evidence from the figure that you're using to answer the question. Like in this case, if I'm told to describe, when I say that as temperature increases, the rate of reaction also increases, come and quote some figures. An example, come to a temperature of 10. Read off the rate of reaction for that temperature, note it down. And then come to a higher temperature, any that you can choose, maybe 20, and still read off the rate of reaction. And when you're writing, include the units. An example, at a temperature of 10 degrees Celsius, the rate of reaction is dash, 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 depending on the units that would have been placed here. Write the units. However, at a temperature of 20 degrees Celsius, the rate of reaction is, you still state it. Now here we cannot because we do not have values on the y-axis. But that is description. As temperature increases, the rate of reaction also increases. Reaching a peak at 40 degrees Celsius, further increase in temperature beyond 40 degrees Celsius, the rate of reaction decreases. So the question is why? Now that takes us to the explanation. Now, at low temperatures, the enzyme, remember we always have our enzyme, assuming this is my enzyme, and it has an active site to which the substrate binds. So this is our substrate. This is the enzyme. This is substrate. So when the enzyme and the substrate are to bind, someone could be wondering, so how do they bind to form an enzyme substrate complex? Remember, they just collide. So they just collide and bump into each other. But by chance, we can have the substrate colliding with the enzyme at the active site. then that will be a successful collision because a reaction is going to occur. But if the substrate comes and collides with an enzyme on this other side other than the active site, no reaction will occur. So these enzymes, as they collide, they move about randomly because they contain or they have kinetic energy. So at low temperatures, the enzymes and the substrate molecules have low kinetic energy. So if they have low kinetic energy, they are not going to be able to collide. They are going to be colliding slowly, and even the chances of an enzyme substrate complex being formed is going to be low. That is why the rate of reaction is low at low temperatures. Enzyme and substrate molecules have low kinetic energy. We have less collisions, less enzyme substrate complexes made. As temperature increases, they gain more kinetic energy. They start moving about more rapidly, more rapidly. They collide. We are going to have more successful collisions, more frequent collisions, and more enzyme substrate complexes made. So when you have more enzyme substrate complexes made, that is how we have the reaction rate increases or increasing. Now, when you look at the temperature of 40, which is the optimum temperature, this optimum temperature, we are having the highest number of enzyme substrate complexes made because the collisions are really at maximum. We have the highest amount of collision between enzyme and substrate. So we have the highest number of enzyme substrate complexes made. That is why we have the highest rate of reaction. The moment the temperature increases beyond the optimum temperature, actually, you can see this temperature has been labeled optimum temperature. So optimum temperature is simply the temperature at which the enzyme is working best. Now, different enzymes have different optimum temperatures. Like our human temperatures, the optimum is 40 degrees Celsius, although your body most of the time keeps the temperature close to optimum at around 37 degrees Celsius, because 37, we shall still have a high rate of reaction. The moment the temperature increases beyond 40 degrees Celsius, the rate just decreases. Why? Because the enzyme molecules gain more kinetic energy. So they vibrate about faster, and the more they vibrate, the higher the chance of the bonds holding them breaking, the hydrogen bonds, the hydrophobic interactions, those bonds that are holding them in their precise 3D shape are going to break. And when the bonds break, the enzyme is going to lose its shape. So the shape of the active site is lost. The moment, let's look at how the shape of the active site is lost. Let's use this illustration, this diagram. This diagram shows you a normal enzyme and a substrate. You can see the substrate has been able to bind to the active site. So a reaction will occur. But the moment the temperature goes beyond the optimum, the enzyme vibrates about fast and it loses its shape. So now here, the active site has lost shape and the substrate cannot be able to bind to this active site. And the rate of reaction is going to decrease because we are going to have less enzyme substrate complexes made. So when the temperature gets beyond 40, usually not all enzymes are denatured at the same time. No, it takes some time for all of them to be denatured, but they start getting denatured one by one. And eventually you can see at a temperature of around maybe 55, almost all the enzymes have been denatured because you can see the rate of reaction is completely 0. So in this case, all the enzymes have been denatured, meaning the shape of the active site has been lost. Substrate cannot bind to the active site. No reaction is going to occur. So that is how temperature affects the rates of enzyme activity. You should be able to describe graphs and you should also be able to give an explanation. So this is what I've been talking about. Some of you would want to make notes. You can simply take a screenshot. But all the information I've been talking about is in this text. Yeah, is in that text. And even right here, that above the optimum temperature, the enzyme's active site is lost. So the shape of the active site is lost. Substrate cannot fit into the active site. Less enzyme substrate complex is made. That is why the reaction decreases. So let's look at another factor that affects enzyme action, and that is pH. When someone talks about pH, we are looking at the measure of the concentration of hydrogen ions in a solution. We could look at the concentration of hydrogen ions in a solution. That is what pH is, standing for potential of hydrogen. And if we have a lot of hydrogen ions, such a pH is usually said to be acidic or a low pH. So there is a pH scale which usually runs from 0 to 14, although 14 is missing here. And usually the pH that is running from around 6.8 below 7, that is usually an acidic pH. Acidic. Then from around 6.8 to around 7.2, that is neutral, a neutral pH. Now beyond that, we call that an alkaline pH. Now pH affects enzymes. Different enzymes prefer working under different pH conditions. An example, in your stomach, you have an enzyme called pepsin. For it prefers an acidic pH. In your mouth, you have amylase. It prefers a neutral pH. In the duodenum, you'll find there are enzymes such as trypsin, lipase. For them, they work best under an alkaline pH. So the moment the pH changes, then it means the enzyme is going to be affected. So how is the enzyme affected? We are going to look at an example of how hydrogen ions affect the enzyme. But before we do that, let's look at this graph. For this graph, we can see that this enzyme works best under an optimum pH. So optimum pH is the pH at which the enzyme is having its highest rate of reaction. And in this case, they are showing us that the enzyme is having the highest rate of reaction, the peak, at a pH of 7, meaning 7 is the optimum pH. So the moment the pH goes away from the optimum, the rate of reaction decreases. The moment it goes away from the optimum, the rate of reaction decreases. Or you could describe it in a way that you mentioned that as the pH increases from 3 towards 7, the rate of reaction also increases. The rate of reaction is highest at a pH of 7. When the pH increases beyond 7, excuse me, when the pH increases beyond 7, we see the rate of reaction decreasing. We see the rate of reaction decreasing. So the question is, how does this pH affect an enzyme? And we are going to look at an example of the hydrogen ions which make the pH to be low or acidic. Remember that hydrogen ions have a positive charge. They are positively charged. And when you look at enzymes, remember, are globular proteins. They have a 3D shape. And that 3D shape is possible because of the bonds that are between the different arar groups of the amino acids that are holding the amino acids that are making up the enzyme. So an example of a bond that is present in enzymes is the ionic bond. You can see that the ionic bond is formed between a charged amino group and a charged carboxyl group. So the charged amino group has a plus, the carboxyl group has a minus. Now, because the hydrogen ions are positively charged, they are going to get attracted to the opposite, which is the negative charge. So we shall have the hydrogen ions interacting with the positively charged arar groups of an enzyme. And once, an example, if this hydrogen ions comes and interacts with this group, it means this bond is going to break. So the major bonds affected are the ionic bonds. So the ionic bonds are going to break. And remember, these bonds are responsible for holding the shape of the enzyme, for holding the enzyme in its shape. So the moment the bonds are broken, the enzyme loses shape, the shape of the active site is going to change, and therefore, the substrate cannot fit into the active site. Substrate can't fit into the active site, you're going to have less enzyme substrate complexes made. And then eventually we are going to have the rate of reaction decreasing. So that is how pH affects enzyme activity. So it is always essential that the enzymes are provided with their optimum pH. And usually in the lab, if you want to provide different pH values, we use what we call buffer solutions. Assuming I want to carry out a reaction, but at different pH. So I'll have maybe one test tube with the enzyme and substrate, but I want that test tube or the enzymes there to work under a pH of maybe 2. And then I have another maybe test tube where I have enzyme and substrate, but I want the enzyme to work at a pH of maybe 4. So for that case, we use what we call buffer solutions. So buffer solutions have a particular pH and they maintain that pH, meaning that to this combination, you will add there a buffer solution of that particular pH you're interested in. So if you add there that buffer solution, like in this case, I'll add a buffer solution of pH 2. These buffer solutions can be made. And that means that the pH in that test tube will remain at 2 until you're done with your investigation. So we can change pH by using buffer solutions. So that is how pH affects enzyme activity. Let's look at another factor, which is enzyme concentration. Enzyme concentration on the rate of enzyme activity. So how does enzyme concentration affect? Remember, with enzyme concentration, we are looking at how much enzyme is present. How much enzyme is present. And we are going to go back to this example which we used. We are going to use an example of enzyme catalyzed. And remember, this enzyme works on hydrogen peroxide. And usually the products are water and oxygen. So I would be interested in finding out, if I increase the concentration of catalase, how is that going to affect the rate of reaction? Will I have more oxygen produced or less oxygen will be released? So we are going to use this example in our explanation. And we are going to use these two graphs. Now, when you look at these two graphs, let's look at part A first. Enzyme catalase can be obtained from nature. An example, within our body, we have their enzyme catalase, like in the liver, because that is where hydrogen peroxide is broken from. Potatoes have enzyme catalase. Celery has enzyme catalase. So sometimes when you want to use enzymes in the lab, we can get them from other organisms. Like you can go get celery, blend it, and then when you blend the celery, you get that extract, the juice from the celery. That juice will be containing the enzyme, and then you can use that enzyme in the lab. So in this investigation, we are changing enzyme concentration. And as enzyme concentration is being changed, so when it came to this investigation, they tried to track the rate at which enzyme catalase works as time went on. They tracked the enzyme activity for 240 seconds. Those are like 3 minutes. Now, in that case, we were having the concentration of the enzyme being changed. The enzyme was obtained from a celery extract. That is why here they're showing you the initial volume of the celery extract. Obviously, if I have less volume, it means less enzyme, because the amount of enzyme in a volume of 0.5 centimeters cubed is not the same as the amount of enzyme in a volume of 1. And it's not the same as the amount of enzyme in the volume of 4. So they got a celery extract and added it to, they got a celery extract and added it to hydrogen peroxide. And then they were able to track the rate of reaction by measuring how much oxygen was released as time went on. Like an example for the test tube where they added a celery extract of this volume 0.5 to the hydrogen peroxide, they were able to track how much oxygen is released in 30 seconds, how about in 60 seconds, and so on. So eventually a graph was plotted. And when you look at this graph, it really shows you that as time increases in both or in all extracts that were used, we see the rate of reaction also increasing because more oxygen is being released. But remember, in the beginning, we mentioned that for you to be able to compare these different concentrations of the enzyme, you have to look at the reaction in the beginning. So it is always important that you consider the first 30 seconds of a reaction so that you can get the initial rate of reaction. So the initial rate of reaction was measured in the first 30 seconds for all the different concentrations of enzyme. Don't forget that whenever you are carrying out an investigation in the lab, only change one factor. Like in this case, we were only changing the concentration of the enzyme. That means that all the other factors had to be kept constant, factors such as temperature, factors such as concentration of hydrogen peroxide, which is the substrate, factors such as volume of hydrogen peroxide, they had to be kept constant so that you're only changing the enzyme concentration. So the initial rate of reaction was obtained. Remember, initial rate of reaction is measured in the first 30 seconds. That is how we have this other graph. So they obtained, like we can see that at 0.5 centimeters cubed of the celery extract, the rate of reaction was, sorry, the initial rate of reaction was around 0.5. And then at 1 centimeter cubed, still you read off the initial rate of reaction. And when you look at this initial rate of reaction, here it was measured in initial rate of reaction per centimeters cubed per minute, but here we have seconds. So someone could be wondering, how come here we have minutes? So they were looking at per minute. That means that, an example, let's look at the initial rate of reaction at 30 seconds for the 0.5 centimeters cubed volume of celery extract. So here, if the initial rate of reaction, if you look at it for this, it's around 0.5. So that means that in 30 seconds, I have 0.5 centimeters cubed of oxygen released. So they had to change this to minutes. How about in 60 seconds? Because 1 minute contains 60 seconds. So that is how they got 60 times 5 divided by 30. And that is how you get the answer, as the answer is going to be 1. So that is how here you see that when they tried to plot the graph of initial rate of reaction against enzyme concentration, we can see that the one for 0.5 corresponds with 1. That is where they got the 1 from. The one for 1.0, still they did the same thing, measured the initial rate of reaction. You can see it's 1 centimeter cubed in 30 seconds. In 60 seconds, obviously it will be 2. So that is how we have 2 here. So they plotted this other graph. This other graph is the best to use, the one that has the initial rate of reaction. Why? Because in the beginning, we are having as much substrate and as much enzyme as possible. So we have more chances of enzymes colliding with the substrate, and then more product being formed. So it is always better to consider measuring the rate of reaction at the beginning, which we call the initial rate of reaction. Now, when you look at this graph, you can obviously see that as the enzyme concentration increases, the rate of reaction also increases, which is so much obvious, because the moment I have more enzyme, I'm going to have more active sites available to be occupied by the substrate. So the more the active sites, it means the more the substrate will fit into the active site, and the more the enzyme substrate complex is made, and that is how the rate of reaction increases. So as you increase enzyme concentration, we also have the rate of reaction also increasing. And this graph shows us that the substrate was in excess. Why? Because we see the rate of reaction is still increasing even with further increase in enzyme concentration, meaning that we have there much more substrate. So this contains what I've already been talking about. It is always best to look at the rate of reaction at the beginning in the first 30 seconds. Because the moment the reaction is underway after 30 seconds, we shall have the amount of substrate varying in the different test tubes that you're using in your investigation. So, but at the very beginning, you have as much substrate as possible. So it is always important that we consider the initial rate of reaction. And yes, those graphs were obtained. I've already talked about that. I've already given the explanation of how these two graphs were obtained. Then we have already seen that the initial rate of reaction increases linearly as the enzyme concentration increases. The initial rate of reaction also increases, because the more the enzyme concentration, the more active sites are available, and the more enzymes are going to be able to bind with the substrates. So the enzyme substrate complex is made. So that is how enzyme concentration affects the rate of reaction of an enzyme. Lastly, let's look at substrate concentration. Substrate concentration affects the rate of enzyme activity. The question is how? So in this investigation, we are still looking at an example of hydrogen peroxide being broken down by catalase. So in this case, the substrate concentration was the one being changed. That means the concentration of hydrogen peroxide. So the concentration of hydrogen peroxide was changed, and they were measuring how much oxygen was released at the different substrate concentrations. And then they measured the initial rate of reaction, just like how we have seen in the other previous example. So they got the substrates, combined it with the enzyme, and then they were looking at how much oxygen is released for over a period of time, every 30 seconds. And then later, the initial rate of reaction was obtained, and we have such a graph that shows us how substrate concentration affects the initial rate of reaction. And in this case, we can see that as substrate concentration increases, obviously we have the initial rate of reaction also increasing. Now we can still see that at low substrate concentrations, an example at this substrate concentration, the rate of reaction is really going to be very low. But at a higher substrate concentration, the rate of reaction is going to increase. So this means that at a low substrate concentration, we are going to have less enzyme substrate complexes made. Why? Because we have less substrates to fit into the active site. So some active sites of the enzyme are going to be left vacant and unoccupied. But when you increase the substrate concentration, it means you're going to have more active sites occupied. And someone could even mention that from this section to around this section before the curve levels off, we are having substrate concentration being a limiting factor, because the more substrates that are added, the more the increase in substrate concentration we see, the more the initial rate of reaction also increases. Meaning that in this case, from this region to around this region, substrate concentration is a limiting factor. But later we see that time comes and the rate of reaction levels off and it becomes constant. So what is the reason for the constant? And this point where we have the highest rate of reaction, where the curve levels off, is what we call the Vmax. Now, Vmax is mainly used if we are looking at the effect of substrate concentration on the rate of enzyme activity. Do not look at temperature affecting enzyme activity and then you mention about Vmax. Vmax, most of the time we use it when we are looking at how substrate concentration affects the rate of reaction. V stands for velocity, max stands for maximum. So that is where we are having the enzyme working at its fastest rate. Why? Because we have very many substrates. Remember, you have been increasing the substrates. So at this point where the curve flattens, all the enzyme's active sites are occupied by the substrate. All active sites available occupied. So the enzyme cannot really work any faster and the rate of reaction becomes constant. Why? Because some substrates are queuing up for the active site, waiting for the enzyme to finish working on one substrate so that they are also worked on. So that is how substrate concentration affects the rate of enzyme activity. Just in summary, remember that in this case, the concentration of hydrogen peroxide, which is the substrate, is the one that was being varied. And then the initial rate of reaction was measured. And we have seen that as substrate concentration increases, the rate of reaction also increases. And why is it that as substrate concentration increases, the rate of reaction also increases? Simply because the more substrate molecules available, the more they are going to enter into the active site, the more enzyme substrate complexes made. Now, if you go on increasing the substrate, but you're keeping the enzyme constant, time is going to come and all the active sites of the enzyme are fully occupied by the substrate. So if you keep on adding more substrates, the substrates start queuing up. So this region where we have Vmax, where we have the curve flattening, is the region where we are having every enzyme occupied. And the substrates are queuing up for the active site to become vacant so that they can be worked on. So that is how substrate concentration affects the rate of enzyme activity. Let's look at this question. They are telling us sketch the shape of the graph, this graph here, that would be obtained if excess hydrogen peroxide were available. Remember, hydrogen peroxide is, that would be obtained if hydrogen peroxide were not available, if excess hydrogen peroxide were not available. Remember that hydrogen peroxide is the substrate. So in this case, we are changing the enzyme concentration. That means that the substrate has to be kept constant, the substrate concentration. but they're telling us to sketch the shape of this graph. How would it be if excess hydrogen peroxide were not present? Because this one shows us that we have excess hydrogen peroxide. What shows? Because we see that the more the enzyme concentration is increased, the reaction just continues to increase linearly. That simply means we have as much substrate as possible present. But if the hydrogen peroxide is not in excess, would expect the curve to increase, the rate of reaction to increase, but then later become constant. Why? Because all the substrate has been turned to product. Why? Because it was not in excess. So we'd expect the curve yes, we would expect this curve or the initial rate of reaction to increase with increase in enzyme concentration, but then later with much more enzyme concentration, we'd expect the curve to flatten. So that is how enzyme is affected by substrate concentration. How enzyme activities are affected by substrate concentration. So those are the 4 factors I wanted us to look at: temperature, pH, enzyme concentration, and substrate concentration. In our next lesson, we shall be looking at some questions, some sample questions, so that we find out how we can apply the knowledge we have learned from this section into those questions so that we are able to excel, because practice makes perfect. So we need to look at as many question papers as possible. See you in the next lesson. Bye.